How's it going? No class yesterday put us behind two lectures, so I figured we'd at least do one of them to finish chapter four. So we're actually going to continue where we left off last week with the first part, and then we're going to do the second part, and hopefully this lecture won't be more than an hour, but we'll see. So please try and watch the whole lecture. You can do it in chunks if it makes it easier for you. It is quite a bit, so I would do it in chunks if I were you. And um, and yeah, that way you can learn and also be able to answer the questions for your homework for chapter four. And then we'll pick up with chapter five on Monday. All right, so um, let's look back at what we were looking at last week. The main gist of it was that we eat food in order to make ATP. So there you have a picture of your energy bar that you're consuming, and that energy bar is going to provide the energy to make ATP. So it's kind of interesting. You probably initially thought that you need the energy you get from food is what you use to do everything you need to do, like move around, for example. But really, the energy you get from food is used to make ATP, which then is used to move around. So, okay, you eat your energy bar that has energy in it and the chemical bonds, and as you break that energy bar down, you that energy is used by the mitochondria to make ATP. So that's at the top there. You're making ATP, that little explosion there of energy. And then that ATP can be used to do cellular work. So again, the moving, that guy riding a bike, that energy that he's actually using to move comes from ATP. Remember, it's the cellular Bitcoin or the currency of the cell. So anytime your cells need to do work, you have to present them with ATP, right? You can't give them, for example, another currency like glucose and ask them to do work. So your muscle cells can only use ATP to do work. And those muscle cells are going to break that ATP down into ADP and use that energy to do stuff, right? And there in the bottom, you have a picture of a endergonic reaction where you're grabbing a lot of blocks and building them together. Um, okay, and let's just review this again. This is the same idea, just in a different form. So ATP formation is coupled with exergonic reactions. Let's look at why that is. So when you produce ATP, when you make it out of ADP and one phosphate, one yellow ball of phosphate. When you're making it, you need energy to make it, right? We looked at last week, we were talking about Lego bricks, and when you put Lego bricks together, you need to put energy into that. That requires energy. So when you produce ATP and you're putting these pieces together, you need energy. So in the mitochondria, you make ATP using energy from food. And that energy comes from an exergonic reaction. That's when you take the food, you're ingesting it, and you break it down, right? And that's what the mitochondria uses to make ATP. So that's why ATP formation is always coupled with exergonic reactions. The exergonic reactions are the reactions that are breaking down food in order to give mitochondria energy to make ATP, right? So if you think about it, the mitochondria are like the translators. They translate the energy from food into energy for cells, ATP. And then ATP breakdown, when you actually break that ATP, that's coupled with endergonic reactions, so the opposite. So let's look at that. When you spend ATP, like if it's currency, right, you're spending it, that's an exergonic reaction. You're breaking it apart, right? And we've seen that typically exergonic reactions release energy. So here you have an ATP molecule. You break one of those phosphates, those yellow balls, by hydrolysis, and that releases energy. And you're left with ADP, which has two phosphates. Um, so obviously you're not breaking ATP down to release energy for no reason. That ATP is going to use it to power reactions that require energy input. So that energy released is used for cellular work. So again, let's look at the same thing in this picture. So ATP formation is coupled with exergonic reactions. You can see there that the energy bar is being broken down. That's an exergonic reaction. And ATP breakdown, when you're spending it, that's coupled with endergonic reactions. You can see there that you're building something. And muscle movement is an endergonic reaction. You require energy for that. Let's look at an example. Let's look at how ATP can be used. ATP breakdown is coupled with exergonic reactions. So a very simple example is when ATP energizes a target molecule, making it likely to bond with other molecules. For example, ATP is a molecule that provides the energy to bring together glucose monomers, right? Those monomers, carbohydrate monomers, glucose, when they come together to form a polymer, they're actually coming together because they're coming together by ATP. So there you can see that ATP donates a phosphate group, a P group, to glucose. And this is a process called phosphorylation when the phosphate group attaches itself to a molecule. So here ATP donates a phosphate group, it attaches to the glucose, and that actually allows the glucose to react with other glucose monomers to form a long carbohydrate chain or a polysaccharide. Another example is when ATP donates a phosphate group that changes the target molecule. So that phosphorylation, that phosphorylation where the phosphate attaches itself to a molecule can change the shape of that molecule. So an example here is a protein, a transport protein. This is a transport protein in the cell membrane that will transport things in and out. And you can see there that the ATP is donating a phosphate group, that yellow ball, and that actually changes the shape of the protein, of that transport protein. So it changes its shape, and when its shape is changed, it releases chloride molecules, in this case, out of the cell. Another example is changing the shape of muscle proteins. So ATP is actually, it phosphorylates muscle proteins called myosin, which changes their shape. And so that pushes the muscle cells, it makes them move. And this is actually how your muscles contract, is by ATP being broken down, donating its phosphate group to a protein, and changing the shape of that protein. In this case, with muscle cells, it changes the shape of the myosin, and that makes that fiber move. Okay. So here is the last question of that first part. Which reaction here is likely coupled with the breakdown of ATP? So think of it this way. The breakdown of ATP, does that release energy or does that require energy? Okay, it releases energy, right? So what is going to happen with that energy? Once ATP breaks down, it releases energy. Is that energy simply going to go to waste, or is it going to be used to power a reaction? It's actually going to be used to power a reaction. So in this case, this A is a reaction that is needing energy. So ATP is broken down and it releases energy, which is going to be used to power this reaction. And this reaction is actually photosynthesis, which we'll learn about next week. Okay, we'll go over this stuff again because I know it's a little complicated, but we'll get it. And again, this is all connected, so you're going to see this over and over and over again, and hopefully it'll make sense. So let's move to the second part of chapter 4. If your mind is a little sore, you can take a break and come back to this later. Okay, so now let's look at enzymes. Enzymes are proteins which speed up biochemical reactions. And these chemical reactions in cells must occur very quickly in order to sustain life. So if you think about it, each cell is performing hundreds, if not thousands, of chemical reactions, and they have to do them super quickly. Think about the Jenga tower, right? So the Jenga tower, if you leave it outside, eventually it'll fall down. But if you knock it over, it's going to happen much more quickly. And that's what an enzyme is doing. It is your hand that is pushing that Jenga tower down, right? And if once that Jenga tower is knocked over, if you want to put it back together, that's going to require energy, and it's not going to happen unless you do it. So same thing in the cell, right? It needs chemical reactions needs to happen really quickly. You can't just leave them to happen on their own. So you need enzymes to do this. So let's look at this ATP cycle that we have and see how this applies. So again, you start with an energy bar that you're breaking down to release energy. So that doesn't just happen on its own. You actually have enzymes in your saliva and in your stomach that break that food down in order to release the energy that's needed to make ATP. And then when you make ATP, that actually doesn't just happen on its own either, right? When you have ADP and a phosphate group that you see there below, when you bring those together, that actually requires an enzyme. An enzyme, it requires energy because the enzyme needs energy to bring those two together. And then once ATP is used to perform cellular work, when ATP is broken down to release energy for reactions that require energy, those reactions that require energy, like your muscle movement and all that, that requires enzymes. So again, in this whole process, enzymes are required to speed everything up so that they can happen fast enough to sustain life. So let's look at what an enzyme is. An enzyme is usually a protein, not always, but it's a molecule that speeds up a chemical reaction without being consumed. So it can be used over and over and over again. So what an enzyme does is it takes a substrate, which is just the initial product or the initial substance, and it binds to an active site, and then it does something to it. So in this picture, we're looking at a substrate that's being broken down. So again, the substrate molecules bind to the enzyme's active site where the chemical reaction occurs. So there, once the substrate is bound to the active site where all the action is happening, that substrate is going to be broken down, right? And again, the enzyme is not broken down. It can be reused over and over and over again. So here's a real life example. Lactase is an enzyme that breaks down lactose. So here you can see that lactose, it's a carbohydrate. It's actually a disaccharide. It has two monosaccharides. And your body needs to break down lactose. It's not good for your body to just have lactose sitting around. So there's an enzyme here that's taking the lactose, the substrate, and breaking it down into its two components, glucose and galactose. Some people can't make lactase. So what do you think happens in those people that don't have lactase? Those are actually the people that are lactose intolerant, right? So if any of you guys listening to this are lactose intolerant, this is one of the reasons why you might be lactose intolerant, is because you don't have the enzyme needed to break down lactose. So it just takes your body much longer to break it down, which is why you feel a little like full and uncomfortable for a long time until that lactose is broken down just naturally. So not all enzymes break a single substrate into two products. Other enzymes combine two substrates into one product, for example. Here's the picture of that, right? So those two products are brought into one by the use of an enzyme. And again, the way that an enzyme can speed up a chemical reaction is by lowering its activation energy. And the activation energy is the energy required to start the reaction. So we saw that the example of a guy riding his bicycle, right? To climb up that hill, you're going to need some energy, and that's activation energy. So there in the graph above it, you can see that there is that little, those little arrows there. That's the activation energy. So in orange, the orange hill, that's the activation energy without the enzyme, and the purple hill, that's the activation energy with the enzyme. It's become much lower. So that just means that you can make this reaction happen a lot quicker because you just don't need as much activation energy, right? It makes the hill smaller, so you can get to the top of that hill quicker and then go down that hill. So only some chemical reactions are occurring in a cell at a given time. Again, you have hundreds, if not thousands, of chemical reactions, but those are very controlled. So the cell is controlling what reactions happen and which ones don't. So how can it control the rate of these chemical reactions? How can it control which reactions happen and which ones don't? There's a few ways it can do this. One is by the use of cofactors, which are simply factors that speed up enzymes or make them more specific. You have inhibitors, which are factors that slow down or stop enzymes. You have negative feedback loops, in which the product of a enzyme is used to slow the enzyme down. And then the opposite, the positive feedback loop, where the product of the enzyme speeds up the enzyme. So we'll look at examples of these, but we'll look at inhibitors first. Cofactors, again, they're just factors that will bind an enzyme and make them quicker or more specific. Inhibitors, though, let's look at those. One way that cells control the rate of chemical reactions is by limiting the activity of the enzymes. So making them less efficient or by stopping them altogether. And inhibitors will make the active site, where all the magic happens, the active site of enzymes, inaccessible. So there's two ways that an inhibitor can do this. It can actually bind the active site, block the active site, so that the substrate can't bind to it. And this is called competitive inhibition because there's competition between the inhibitor and the substrate. Then there's also non-competitive inhibition, where an inhibitor will bind the enzyme and change the shape of the active site. So there you can see that the shape of the active site has gone from a square to a triangle, so the substrate can't bind there anymore. So by using inhibitors, a cell can prevent certain reactions from happening, right? Then there's a positive feedback mechanism where the product of a reaction stimulates its own production. So here we have a chain of enzymes, 1, 2, 3, 4. And there's a substrate A which is modified each time by the different enzyme until you get the final product there in brown. So that product, though, can actually fit into that first enzyme and make it go faster. So again, this is like a cofactor. It makes the enzyme more efficient. So it's a positive feedback because every time you make a product, it's encouraging this process. It's encouraging turning substrate A into the product, right? So it just makes it go faster and faster and faster. That's why there's a loop there. It's a positive feedback loop. And an example of this in our bodies is the initial stages of childbirth. It releases a molecule called oxytocin. It's known as the love molecule. So, anyway, you start childbirth, you release this oxytocin, and oxytocin then stimulates childbirth. So, it's this like positive feedback loop, right? It'll make it go a lot faster. The opposite process is called negative feedback. So, this is where the product of a reaction slows down the production of more product. So in this case, it's the same chain, except in this example, the product is going to bind to the first enzyme and slow it down, inhibit it, right? So in this case, the product is an inhibitor. In the positive feedback loop, the product is a cofactor, which speeds up the enzyme. In the negative feedback loop, the product is a inhibitor, which will inhibit the enzyme. So an example of this in your body is when your cells make a ton of adrenaline. So they make adrenaline, but adrenaline at the same time makes your brain release hormones that stop the adrenaline production. So the more adrenaline you release, actually, the more you stimulate your brain to stop adrenaline. Otherwise, adrenaline is going to stay active for a long time, or you're going to keep making it, and that's just not good for your body. It can cause you to crash, right? Because your body's using, once adrenaline is released, your body's using a ton of energy. So the way your body stops making adrenaline is because adrenaline tells your brain to make hormones to stop adrenaline production. So again, here I just want you to realize that your cells have different ways of controlling chemical reactions, right? They can speed them up by using cofactors or positive feedback loops, or slow them down using inhibitors or negative feedback loops. Temperature also affects enzyme activity. So most enzymes have an optimal temperature at which their activity is greatest. So here, the enzymes in our human body function most optimally at body temperature, which is 37 degrees. Whereas the enzyme from a bacteria that lives in a hot spring, because the temperature that bacteria lives in is super hot, the optimal temperature for for the enzymes in that bacteria is going to be way higher. And they also have optimal salt concentrations and pH concentrations at which they function most quickly. So your cells can also regulate, or that's why your cells actually want to maintain an optimal temperature and salt and pH concentration, is so that enzymes can be functioning optimally. Okay, so here's a question. According to the graph, what temperature do you predict the bacterial enzyme becomes denatured? So we saw that in the last unit, that denatured, a protein that becomes denatured loses its shape, and so it can't function anymore. So the question is, at what temperature is the enzyme from this bacteria becoming denatured, losing its activity? So here on the y-axis, you have the rate of the reaction, basically how efficiently the reaction is happening. And you can see that the peak is at 70, in the high 70s. But then once you increase temperature past that, you lose that enzyme activity. And that's because the enzyme is being denatured. So what temperature is that? 78, right? 73 degrees might be the optimal temperature, but at 78, boom, you lose activity because you denature the protein. All right, so now let's change gears and look at how your cells transport things inside and out. So we kind of looked at this when we were looking at the plasma membrane, at the cell membrane that's made up of a phospholipid bilayer. And here's the image of it, right? And this phospholipid bilayer allows the interior of the cell to be different from its exterior. And it maintains this difference by regulating the transport of molecules across its membrane. So it controls what goes in and out, right? And there's two types of transport in cells. There's passive transport, which is a type of transport that does not require energy, which is why it's passive. When you're a passive person, that means you're not exerting a lot of energy. And there's active transport, a type of transport that does require energy. And this is where enzymes come into play. So this is linking what we just learned about enzymes with the stuff we learned about the cell membrane, this bilayer, this phospholipid bilayer. Okay, so let's start with passive transport. Passive transport does not require energy. It is driven by concentration gradients. We'll learn what those are in a second. But there's three types of passive transport: simple diffusion, osmosis, and facilitated diffusion. I really want you to understand the first two. So a concentration gradient occurs when there are areas of high concentration and areas of low concentration in a solution. For example, when you're preparing tea. So here you have, so say you have your mug with your hot boiling water and you insert your tea bag. As it starts to diffuse, you automatically have areas of high concentration of tea and then areas that have no tea at all. So areas of low concentration. And that tea is going to diffuse through the water, through the boiling water. And what's happening is that it's moving from areas of high concentration to areas of low concentration. That's called moving down its concentration gradient until eventually the whole water is diffused with tea, right? It becomes tea. So again, particles naturally move down their concentration gradient. This is a natural thing. You don't even need hot water. This can happen with room temperature water. That tea will eventually diffuse into the water. And the reason that is because of entropy. So remember, entropy is the tendency toward randomness and disorder. So when you put a tea bag into the water, that's not a random system. There is like a really high concentration of tea in one spot, and the rest of it has no tea at all. That's very structured. It's the universe likes it a lot better when there is an equal distribution of stuff, right? When it's all random. The fact that all the tea is in the tea bag in the boiling water is non-random. For the tea to diffuse everywhere, that is random. So again, entropy is what's driving this diffusion of stuff from high concentration areas to low concentration areas. So in simple diffusion, particles move from high concentration to low concentration. Again, that is, they move down their concentration gradient. So here in the picture, you have an area of high concentration, say that is inside of the cell, and then you have an area of low concentration on the other side of the cell membrane. So substances will move across the plasma membrane or the cell membrane naturally because of entropy, because it's trying to get to an equilibrium, right? Again, think of entropy as equilibrium. The universe likes equilibrium, it likes randomness. So here, the particles are going to move from the area of high concentration to the area of low concentration. And this only happens with small nonpolar particles, right? Because we saw that the lipid bilayer, the core of it is hydrophobic. So it hates interacting with things that have a charge, because that core, that hydrophobic core, are these nonpolar tails. It's these tails that do not have charges. So they don't like to let things that have a charge go through. They don't like to let polar things through. The only things that will naturally go through the cell membrane, diffuse across the cell membrane, are small nonpolar particles, right? Things that don't have a charge. Okay. Osmosis is very similar, except that it's specifically talking about water. So osmosis is simple diffusion, except we're only talking about water. So in this picture, on the left, you have this U-shaped tube, and you have a selectively permeable membrane in the middle. So there in the zoom in, those purple lines, that's indicating there's a selectively permeable membrane, which we saw that the lipid bilayer is a selectively permeable membrane. It only allows certain things to go through. Well, this membrane will only allow water to go through, which are those little red and white molecules, right? The H2O. So those water molecules can diffuse freely across that selectively permeable membrane. But there's solutes in there where the blue and green molecules. So in the picture, if you go back up to the left, there's a higher concentration of solute on the right. So water is actually going to go, which means basically that there's more water molecules on the left than there are on the right, because the right has a higher concentration of the other particles. So water is going to naturally diffuse through that membrane toward the right, where all those arrows are pointing, right? They're pointing to the right, because it's going to try and find equilibrium. So now if you look at the right, you see that the water level has risen on the right side and it has dropped on the left side. So this actually, you can do this at home if you have the materials, but this is a really simple and cool experiment where you can actually move water and cause water to go up on one side simply by having a higher concentration of solute on one side. So after the water level has risen after you have more water on the right side and less water on the left side, now you actually have equal concentrations of solutes. So you have equal concentration of water and equal concentration of solute. So this is the same principle that we saw at work when you were looking at your plant cells in our last lab, and you put them in a solution where there were no salts in deionized water, and in a solution that had a lot of salts, right? Salt water. And you saw that when you had water with no salts, a hypotonic surrounding, those cells brought in water. That water entered the cell so that it could actually try and find equilibrium between the amount of solute inside the cell and outside the cell, right? And so those cells got large and rigid. Whereas if you put those cells in a solution that had a lot of salt, that water is actually going to leave the cell to try and dilute the solution with a lot of salt and try and equilibrate the concentration of solutes. And so those cells shrink and actually wilt, right? So that's why if you add a lot of water to plants, they are nice and healthy. And if that water starts to evaporate and it collects a higher concentration of salts and stuff, that plant is going to wither. That's why you should change the water of flowers and plants regularly so that they stay rigid, because that water is going into the cells. Alrighty, now charged particles that have a charge, we saw that those particles cannot go through the plasma membrane. They can't go through the cell membrane because that hydrophobic core, that nonpolar core, will not let particles that have a charge to go through, right? They're very inclusive. So these particles must use facilitated diffusion to move across the cell membrane. So facilitated diffusion is just a transport channel, that purple channel there, that will allow these charged particles to move through the plasma membrane or the cell membrane. And again, facilitated diffusion is passive, so it doesn't require energy because the particles move down their concentration gradient. So this is similar to diffusion in that the particles are moving from an area of high concentration on the right to an area of low concentration on the left, right? But they need this protein channel, this hole, so that they can move through the cell membrane. And again, the hydrophobic tails of the phospholipid, that hydrophobic core, the middle of the membrane, will repel hydrophilic substances or charged substances. Therefore, ions such as chloride and sodium ions can't go. They will not go through the membrane, so they need this transport channel to go through. And polar substances also have to go through this protein channel to cross the membrane. But again, this is passive transport because it's moving down a concentration gradient. Okay, so let's revisit. So you have two types of transport: passive transport that does not require energy, and it doesn't require energy because particles move down their concentration gradient, right? And this is just anytime there's a concentration gradient, particles will naturally move down the gradient because of entropy. So you don't need energy for that. That's actually driven by entropy. Now there's active transport that does require energy, and this is because this happens when particles are moving against their concentration gradient. So they're moving the opposite way. So again, that's going against entropy. That's going against this randomness in the universe, or this randomness the universe wants to attain. So you actually need to get to use energy to do this. So this is active transport. And this is when the cell uses energy and a transport protein to move a substance against its concentration gradient. So you see here that the movement is going from low concentration to high concentration. So it requires a protein and it requires energy. So it requires ATP in order to pump. Literally, this protein is pumping the molecule against its concentration gradient. So here's a question to try and put this together, bring this together. This is a sodium-potassium pump, which is pumping, in this case, sodium outside to the outside of the cell, and it's pumping potassium, which is K+, into the cell. So the question is, why does this sodium-potassium pump require energy? So refer to the image below. And what you want to look for here is a concentration gradient. So here you're seeing, if you look at the first and second panel, that the pump is moving these purple balls, the sodium, from a region of low concentration, which is in the cell, the cytoplasm, to the outside of the cell, which is a region of high concentration. So it's moving against its concentration gradient, right? So if you look at the third and the fourth panel, what it does then is it takes potassium, which are these yellow squares, from the outside of the cell, which has a low potassium concentration, to the inside of the cell, which has a high potassium concentration. So again, it's moving potassium against its concentration gradient. And you can see that it uses energy because in that first panel, it's using ATP to do this. So why does it require energy? Because it's moving these solutes, these molecules, against their concentration gradients. All right, hopefully that's clear. We'll go over this again in class because it helps to do this in person. This is tough stuff. But you guys are brilliant kids. You guys had the highest scores out of any of my 3 classes, so I know that we'll be able to understand all this. Okay, so large substances, so far we've been looking at small substances. Large substances enter or leave the cells in vesicles, which are little membrane bubbles, basically. So here you have endocytosis, which allows a cell to engulf fluids and large molecules and bring them into the cell. So there you see the inside of the cell, the cytoplasm is in blue, and the outside is in tan. And so you see in the first panel that you have all these substances, these large molecules that need to be imported into the cell. So what it's going to do is that plasma membrane or the cell membrane is going to bud inward, and it's going to start budding inward until it traps these particles and it closes off and forms a vesicle. And then this bubble is going to bud off and go into the cell. So that is how cells bring in large amounts of stuff or large particles. is by endocytosis. And again, remember the keyword endo means into, and cytosis is like the cell, right? Into the cell. Cyto means cell. The opposite thing is when the cell needs to release a lot of molecules or a lot of large molecules, then it does the opposite process called exocytosis. So here on the first panel, you have a vesicle with a bunch of particles, and that vesicle is going to touch the cell membrane and fuse with the cell membrane. It's going to merge with the cell membrane and release all of the contents to the outside of the cell. So this is what's happening when your adrenaline glands are releasing adrenaline, or your milk glands are releasing milk, or your brain is releasing hormones, right? It's using this process called exocytosis. All right, so let's summarize what we just learned. So the first question, this is basically going to be a tree that's going to help you understand all of these ways that the cell moves things in and out. And the first question is a substance that the cell is trying to move in or out very large? If the answer is no, the next question is a substance moving down its concentration gradient from a region of high concentration to low concentration? If it is, then the next question is a substance nonpolar? Or in other words, does it not have a charge? If it doesn't have a charge, then this substance is moving through the cell membrane using simple diffusion. So this nonpolar substance is simply moving from an area of high concentration to an area of low concentration across the cell membrane. If the substance is not nonpolar, so if it is polar and it has a charge, then it's moving using facilitated diffusion, namely a protein channel that allows this charged particle to move from an area of high concentration to an area of low concentration. So both of these methods are passive. They do not require any energy. If the substance is moving against the concentration gradient, then it requires active transport. So again, the question is a substance moving down its concentration gradient? If the answer is no, then it's needing energy, and that's active transport. And that's where using energy to literally pump a particle or a substance against its concentration gradient from an area of low concentration to an area of high concentration. And then if the substance is very large, then depending on whether it's entering or leaving a cell, if it's entering, you're using endocytosis to bring it into the cell. Or if it's leaving, you're using exocytosis to make it leave the cell, right? So this hopefully helps you learn how these things are related. But this is all about how cells move things in and out of the cell membrane. And the key thing here is that enzymes are being used in active transport to pump things across or against their concentration gradient. Okay, so last question, and we're almost done. In this picture, you have a protein channel, and you have a solute that is moving from a high concentration gradient to a low concentration gradient. So what it's asking here is that it's first asking whether the solute is polar or nonpolar, and then it's asking you whether the transport requires, whether it requires or doesn't require ATP. So the first way you can tell whether it's polar or nonpolar, if it's something nonpolar, if it doesn't have a charge, it's not going to need a protein to move through the cell membrane, right? So in this example, this substance needs a protein channel. So that means that this is a polar substance. It has a charge, and it's not going to be able to move through that cell membrane without this channel, without this protein channel. And then the second question is energy related. We saw that if particles are moving down their concentration gradient, they're not going to require energy. If they're moving against their concentration gradient, then you do need energy to pump that particle against the concentration gradient. In this example, you're moving from high concentration to low concentration. So it's moving down the concentration gradient, so it doesn't need any energy. So the right answer is the solute is polar and its transport does not require ATP. Okay. So I know that was a lot, but I hope you enjoyed it. Again, we'll go over this stuff on Monday. I will assign homework, though, on Chapter 4, so please, please do it. Again, each homework is worth 10 points, and believe me, it adds up. I've seen students who did excellent on exams and ended up getting a low score, their final low score, because they didn't do homework, and students who did poorly on exams and the homework actually bumped them up to a decent grade. So please do the Chapter 4 homework, and I hope you guys have a great rest of the week. Hopefully no one is suffering because of the ice and the snow, right? If anything, you guys didn't have to come to school, so that's a great thing. And who knows, maybe tomorrow your lab will be canceled too. We'll see. All right, hope you guys have a great one, and I'll see you guys on Monday. Bye.